right? That involved the innate things like phagocytosis, inflammation, barriers, and then, it says adaptive up there. That's another word for it, or specific is another word. And then there are two broad categories under acquired. There's humoral, which involves making antibodies, and then there's cell-mediated, sometimes it's referred to as, or cellular. Right, this involves B cells, and this involves T cells. And we're going to go through that in this section. Now, you do in this, right, it says that the adaptive or acquired, right, it does enhance the non-specific, right? That means, inflammation can be, right, started, or phagocytosis can be enhanced. So there's some crosstalk. And I hope, right, this concept of memory cells, both B cells and T cells, when they get activated, right, so a B cell has an IgM receptor to bind to an antigen. T cells have the T cell receptor. Okay. So these are they call them receptors for the antigen. If the antigen sticks, then the B cell turns into plasma cells in response to the antigen. The plasma cell makes antibodies. We say the T cell turns into, I like to use cytotoxic T lymphocyte. Cytotoxic T lymphocyte, CTL, is an activated T cell. Now, during the activation process, memory cells are also generated. So you start off, say, with one B cell that has one receptor. When it gets activated, then it turns into clones of itself, and there might be 1,000 of them. Some are memory, some are plasma cells. CTLs, we're going to keep it real simple, right? They turn into many cells as well, from one cell to many, and some of them are memory cells. They're both lymphocytes, but these are the two broad categories. Right? You get a memory. Now, you don't respond to antigens. This process doesn't occur until you're about 6 months old. So until then, you rely on your mother's antibodies and breast milk, or what crossed the placenta while you were still inside her. We'll talk about that. And then your acquired immunity, your innate immunity. So non-specific in this sense refers to the innate, things like phagocytosis. The lymphocytes, the key. Lymphocytes, we're going to call them secondary lymphoid organs, is another name that's used. They hang out in lymph tissue, fire patches, spleen, tonsils, and they're waiting for a foreign antigen, right? And then they respond. Those are secondary lymphoid organs. The primary lymphoid organs are what we call the places where lymphocytes become either B cells or T cells. So before they're a B cell, the more immature, right there, they're generated continuously in the bone marrow. Right, you're making white blood cells. And then they hang out in the bone marrow, and that's where they end up being a B cell if they turn into a B cell. A B cell is identified by having the receptor for the antigen, right? This IgM. That makes it a B cell. A T cell matures in the thymus, and they call them T cells because they have the T cell receptor, and that's the receptor that binds to antigen. Right? Think of them as locks. Right? There's 100,000 B cells with 100,000 different locks. There's, say, 100,000 T cells that vary by the lock found on the T cell receptor. The keys are the antigen. So I just throw out this number. It's an approximation, just to give you an idea of the ballpark figure of how many different antigens our immune system can respond to. Right, there's many of them. And they don't, not all the B cells, say if there's 100,000 of them, not all of them are going to be activated because you're not going to be exposed to all the antigens that they are capable of responding to. I'm not sure the number that are. I'm sure somebody's figured that out. It's quite a few. So, like as I put on the board, there's humoral. That refers to anything antibody. Cellular T cells that are seeking other cells. That's what it's called, cellular, because it's our cells killing our cells. They're only going to hopefully do it if it's infected with a virus or infected with bacteria. Right, that's the antigen-specific nature of it. If a cell contains the antigen on its surface that this T cell was activated against, then it will destroy that cell. Now, an antigen, it's kind of complicated, right? If you have a big protein, and like say there's 1,000 amino acids all linked together on this line. You can think of it as a crossword, well, that wouldn't be a good word. A puzzle piece, I use that example too. You can think of it, if you had 1,000 puzzle pieces, each puzzle piece has a different shape. I think that's kind of how you want to think of antigens, right? So there's 100,000 different shapes that can fit those receptors. There's only of a protein, only about 4 or 5 amino acids make up the puzzle piece. That's the antigen. The epitope is another word for it. It's the antigenic determinant. That's the very specific part. An immunogen means that the piece, the small piece, has to be in the context of a bigger piece. And that's just the way it works. And I put the bacteria up here. And if you think about it, the only way you're going to recognize these antigens, if you make an antibody and you want the antibody to bind, it's got to be on the surface of things. Right? It's got to be on the surface of a virus. It has to be on the surface of, at least for the antibody to get to it. Right? If these antigens are in the middle of the bacteria, the antibody can't get through the bacteria. It's not going to be helpful. So there's 3 different antigens. They show them as simply 3 different shapes. But you wouldn't expect the antibody A that binds to the triangle to bind to the other two or any other antigen. Now sometimes it happens, rarely. You wouldn't expect antibody B that binds to the semicircle to bind to the other two either, right? They have to be specific or else the whole thing doesn't work. Now this concept of a hapten, you know what I was saying, the antigen or this small piece needs to be on a large molecule, like a protein. So let's say this is a puzzle piece. They call them haptens because they're too small to be recognized as an antigen. So they're non-antigenic, right? An antigen is something that elicits B cells or T cells. There's a response. There's no response to things like haptens unless they attach to a larger protein. Normally it's protein most of the time. It just got to be in the big. These guys know the difference when they recognize them. Then it becomes a complete antigen. And it becomes antigenic. But haptens are not, right, by definition. Now when we talk about penicillin in section 5, if you have an allergy to penicillin, it's because penicillin acts as a hapten and can stick to proteins on our body, and some people are allergic to it, and we'll kind of go through that example then. So immunogen or immunoglobulin, there are 5 of them altogether. I think there's even more than that, right? There's an A version of it, there's a G version, there's an M version, E version. These are 5 different proteins. There's 5 different immunoglobulin proteins. All 5 of them have this general structure. That's how they look. But there's little differences in the amino acids in them. They serve a purpose, right? They bind to antigen. They have what's called the, I refer to it as a lock. And it's a variable region because those amino acids have to have different shapes to bind to 100,000 different antigens, right? That's how they vary. The spot. Even the TCR, when we get to that receptor varies so that it can bind to 100,000 different antigens. So the constant region stays same for each one of these, right? There's Ig, immunoglobulin A, there's immunoglobulin G, there's immunoglobulin M. So what we'll do is I'll explain like IgG, IgM are found in the serum, IgA is found in, like say, secretions. But they're all made by plasma cell. When the plasma cell is activated, then it has to decide which one of these 5 it's going to produce. But every time it produces IgA, it binds to the same antigen that started the process. Or if it says, okay, now I'm going to make IgG. This IgG and IgA made by the same plasma cell will bind to the same antigen, right? Because that remains constant. So we usually use the Y shape. That's why we use Y. It's not, the textbooks started drawing it like that, but it's technically, these are 2 proteins, and they kind of curl up together in the middle. And then the blue is this part, it sticks out over here. And they call it a hinge region because it's flexible. This is called an FC region and FAB. There's a complement binding domain because complement, or C1, that protein we discussed in the section 13, right, can bind to that portion of IgG and IgM. So antibodies stick to things. Specifically, if they can agglutinate bacteria, which means cause them to come together because they have two arms. IgM actually has 10 arms. And they can agglutinate red blood cells. When you type blood, you can see them coming together that way. We talked about oxidization and enhancing phagocytosis. Neutralization is when they bind a virus or even toxins. And when they do that, it prevents them from binding to whatever they need to bind to do whatever they do. Right? Viruses bind to receptors on host proteins or host cells. They can't do that if they've got virus or they've got antibody attached to their spike proteins. Then we talked about complement in section 13 too. There's this ADCC as well. It's, don't worry about understanding what that is. It's a combination of cell-mediated, cellular, and humoral. Antibodies are involved. So as I put on the board, there are 5 different types of antibody, which the plasma cells that are generated, they're going to decide which one of the 5 to make. They're always going to direct those antibodies to bind to the same antigen. Right? The variable region is dictated by whatever IgM, right, say it binds to the COVID-19, right? COVID-19 comes along and sticks to it and activates this B cell. That generates plasma cells and memory cells. It's going to decide to make either an IgA directed against COVID-19. It might start making IgG first against COVID-19. It might do that for a few months and go, okay, now I'm going to start making IgM against the COVID-19. It's going to be the same plasma cells, and they just have to decide, right? It's dictated, in their DNA. So if you make IgA, you actually put 2 Ig molecules together. If you just say the word Ig, then you're referring to this. You're not saying which one of the 5 it is. Okay? So there's 2 of them stuck together here, and when plasma cells make IgM, there's 5 Igs. They can bind to 10 antigens, 1 molecule. If you have IgM, don't confuse when a plasma cell secretes IgM with when IgM is used as a receptor. They're 2 different scenarios. I'm even going to describe IgE in section 15 on mast cells. There's what's called an Fc receptor that binds to, or IgEs bind to that on the mast cell. So an IgE is secreted by a plasma cell and it ends up on mast cells. Then once that occurs, then it acts as a receptor on the mast cell because it can bind the antigen that caused this plasma cell to start, right? Whatever started the process. Now, when an antigen or an allergen binds to this, then the mast cell is activated or releases histamine that causes allergies. Now, I only bring this up just so I can bring up the fact, right? You have IgE on a receptor here. You have IgM as a receptor here. Right? Don't get them confused. If you secrete IgM from a plasma cell, right, it has nothing to do with this receptor that's always found on B cells. Right? B cells don't secrete antibodies, plasma cells do. They're cousins. That's why I put allergic reactions under IgE. We don't really talk about IgD for anything. IgM is found in the serum. IgG is found in the serum. You should remember IgA is a dimer, it's called, because there's two parts to it, two IGs. Right? Pentamer means there's 5. You find IgA, it has a secretory component. So clonal selection is the term that's always been used. I tried to help you understand this, right? You have 100,000 B cells that have 100,000 different locks. They're showing 6 of them up here. And they're trying to make the distinction on the receptor by drawing little different shapes on the receptors to help you see that. It's very simplified. But an antigen that binds to this receptor will not bind to the others. So clonal selection means when this cell is activated by the antigen, the B cell, then it becomes many cells. Memory cells. All of the cells now that came from that one clone will specifically bind, like I said here, with COVID-19. Then regardless of the antibody that the plasma cells that are generated, the thing they all have in common is they'll stick to the same antigen, whatever antibody they make of the 5. It's a difficult concept to wrap your head around, right? The clones have the option of making different types, but they all bind to the same antigen. So you expand the B cell, it goes from being 1 to many. The memory cells that are generated could last a lifetime, but not that you don't really know. If a cell to last 50 years is a pretty long time for a cell. So the activation results in plasma and memory cells. And this is a pretty complicated look at the process. I've got a better one I'm going to show you in a minute. But and the antigen presenting cell is the B cell, actually in this context. And it presents to a T helper, and then the B cell is activated by the antigen. That generates many cells, right? Including memory cells. Now the way it works is the B cell, it'll stick to the receptor, and then it gets internalized into the B cell. And don't worry too much about that process. Unless you're a cell biologist, it's really difficult to understand it all. But the receptor is in the cytoplasm, and then it makes its way out to the surface. It's called the MHC2. I've got class 2 there. You might just see MHC2 subscript instead, right? There's also MHC1. which is class 1. We talked about this one in the innate immunity. I said I associated it with a social security number and being tissue type. MHC2 is also used in this context, so I'll mention it again in a minute. But when you activate the B cell, the MHC2 is used. It holds the antigen and presents the antigen to a T helper cell. The T helper cell has a TCR that's hopefully specific. If it has the correct shape, the antigen, then the T cell activates the B cell, making memory cells and plasma cells. So when we activate a B cell, to summarize, right, the APC stands for antigen presenting cell. The B cell is used. It uses the MHC2, which can bind to any type of antigen. The T helper cell has the TCR, which is got to be specific. So you have two specificities. You have a specific antigen for the IgM. It's like a lock and key thing. When you present the antigen, MHC2 is used to present any antigen. So it's not specific. It just holds it and presents it to the T helper. But when the T helper has specificity for whatever antigen, right? As I said before, the T cell receptors, there's about 100,000 different receptors that have the ability to bind 100,000 different antigens. So you have the memory of the exposure just means you've generated memory cells. So this is a typical curve that's used. If you look, antibody, the amount of antibody found in your serum, as it goes up, the line goes up. This is a time period, right? This is about 10 or 14 days. And then within 1 or 2 days, you get this curve. Now, when they say, you no longer have, let's say, you get a vaccine, you generate antibody and the antibody goes away, right? And then they argue, well, you need another booster. Why? If you get, most of the time, 99% of the time, as soon as you're exposed the second time, naturally, instead of with a shot, the antibody titer is going up. So case in point, why are they making this booster thing, such a big deal? Other than it makes some companies billions of dollars. So that's the problem with acquired immunity, right? I like to just use the 14-day time point. It could be 10 to 14 days or something like that. It's different for different people. But the first time you're exposed, it's going to take you about 14 days before you start seeing the antibodies being produced and found in your bloodstream. But the good news is then you're immunized. You have immunity. Now when you say, what is immunity? You can define it, there's connotations, right? I've been exposed to something and now I have memory cells. That could be one, right? You might say, well, I'm immune because I don't die from something now. Like with the children, right? And COVID-19. They weren't dying from COVID-19 unless they had they had cancer or they were really obese, I think was the other. That's what we mean. But in the truest sense, immunity means you've generated memory cells because you've been exposed to something, and that's the whole purpose of vaccination, right? Is to make memory cells. It's not how long you continue to make antibody after that just depends on the person. And normally, when you evaluate a vaccine, you say if it's 90% effective, then 90% of those that were vaccinated are making antibodies now. If they don't generate antibodies after vaccination, it's because the B cell didn't have the appropriate receptor and you don't see antibodies being produced. So if you get vaccinated a second time, you're probably not going to help either. Right, that's kind of how that works. Or your immune system isn't working for some reason. Now we recall these MHC1 and MHC2, they're receptors, but they're non-specific, right? They just hold pieces of protein, and that's what most antigens are, right? In the MHC1, there's a little spot that holds the antigen. It just sticks to it for some reason. I don't know why. The MHC2 kind of cups it, you know, like, it looks like claws on a crab. And so antigen presenting cells, you can present all different types of antigens using either one or two. I just explained with the B cell that the MHC2 is involved for presenting the antigen. When we do the T cells, T cytotoxic, then I'll mention MHC1. Now this is a depiction, I usually at this point I try to emphasize, we're going to go talk about T cells now. The T cell receptor is very similar to the IgM of the B cell because it's a receptor. It has the ability to specifically bind 100,000, just like it has a variable region. The T cell receptor has to have a variable region as well. So this is a cartoon. This is the protein. I don't know how they get that shape, but it's a protein, and they put the V there to say there's a portion of this protein that varies so that you can have, say, 100,000 different locks for 100,000 different keys that would fit each lock. This one's specific, this is specific, these are not. So what does cellular immunity do? It does a few things. And a couple of them really don't involve antigen recognition, but they're included here as well. Now when you kill an activated T cell kills a virally infected or bacterially infected cell, it's only going to do it if those infected cells have a pathogenic antigen that activated the T cell. That's why they're going to do that. Now when you reject tumor cells and transplantation of tissue, that might not be the case. But there are a lot of acronyms in immunology, and I'm not really getting into all of them. But like, for instance, CD4 and CD8 are commonly used to describe two major types of T cells, right? There are T helper, or TH, and there's T sub C for T cytotoxic. Right, so you could say T cytotoxic or T sub C. You could say T helper or T sub H to denote either one. If you're a T helper or a T cytotoxic, both of them have the T cell receptors. That's what makes them the T cell. What makes them one or the other is that CD8 is a marker found on lymphocytes that make them. Or CD4 is another marker. Now when we activate the T helper, we're going to do that in a minute. I just put this here to help you with that, right? MHC2 is the antigen presenting molecule. When we activate the T cytotoxic cells, the antigen presenting cell uses MHC1. That's what we're going to do now. Right? How are the T cells activated? We kind of went through the B cell already. Right? Dendritic cells and macrophages are used as the antigen presenting cell. So macrophage is a phagocyte. Right? It can phagocytose bacteria, and in the process, a piece of the bacteria gets into its MHC2, and that antigen then is presented by the macrophage to the T helper. So in this case, the little peanut, if you can see that, is the antigen, and the MHC2 is kind of grasping it, and it's looking to see if the TCR on the T helper, if it fits in the receptor. The key is the peanut or antigen, and we want to know, is the key fit the lock? If it does, then the T helper is activated. All right. Now, we get T memory cells in the process, right? We start off with one cell, and they're showing 5 different cells, or 6, right? There are many of them. We're not getting into all of them. You go from 1 to hundreds to, I don't know if it's thousands, but it's probably over 1,000. The T helper 2 is what's needed to activate the B cell, which we already did. But before the T helper could help activate the B cell, it had to already been activated by the antigen that's now going to be activating this B cell. Right? So there's crosstalk, right? You can make antibodies against COVID-19. And you can also generate T helper or T cytotoxic T cells that will look for, like in this case, right, the antigen on the B cell, and that helps it become activated if it's a T helper. A T cytotoxic cell, which we're going to go into next, right, it gets activated by COVID-19, and then it's looking for cells that are infected with COVID-19, and it'll kill them. We'll do that next. Any questions? Get a little complicated. But just to go back real quick, right, all I'm saying is we jumped ahead of the ballpark, right? This T helper cell, it had to have been activated previously to the only way it's going to, right, help the B cell is if it was activated by the same antigen that now is activating that B cell. And I'm just explaining how the T helper gets activated, right, at this point. Now, we did the T helper. Now we're going to activate the T cytotoxic. Now, for simplicity purposes, I'm saying the activated T cytotoxic cell becomes the cytotoxic T lymphocyte, CTL, when it's activated. I don't know if that's really clear in textbooks, but I'll make that distinction for this class. So we have the same scenario as we did with the T helper. The antigen presenting cell can be the macrophage or dendritic, but now it's going to use the MHC1 it's like a one-handed, it's hanging onto it with just one hand, the peanut. And if the T cytotoxic MHC, or TCR rather, if the antigen fits that lock, then the T cytotoxic becomes a cytotoxic T lymphocyte or activated T cell. Now the reason what it does, I guess, right? It hangs once it's been activated. Then, if that T cell ever encounters a cell that was infected with the same pathogen that activated it, has the same antigen, and that antigen is expressed on the surface of this host cell, it'll fit the T cell receptor, and then this host cell is destroyed by the cytotoxic T lymphocyte, right, by using porphyrins or enzymes that break it down. That's what cellular immunity is. Right? It's not so much the T helper as it is the T cytotoxic going after cells that are infected with virus or bacteria. So it kills this cytotoxic T lymphocyte, kills cells that are infected. Only those cells, though, that have the antigen on its surface that originally activated this cell, that are infected with a virus or bacteria, like I said before, right? That's what we're describing. They are also involved in rejection of tumor cells, but not antigen-specific or transplantation rejection, which I think I mentioned was involved MHC1 not having the right social security number. But this is strictly the antigen specificity. Now, NK cells are part of cellular immunity, but they are the innate immunity. Right? It's like killing cancer cells, but they're not killing based on antigens. Right? They're not, the reason you say it's innate, it's the cellular immunity that, right, is not looking for a foreign antigen on the surface. It's just killing random indiscriminately. Right, we make memory cells. We talked about CD4, CD8. When the antigen presenting cells, or all the T cells, whether it's T helper or T cytotoxic, can be dendritic cells or macrophages, and that's because they're involved in the elimination of bacteria and virus. You know, when a macrophage phagocytizes a bacteria, then pieces of that bacteria end up on the surface of that macrophage, and then that macrophage could encounter, you know, say like this, the macrophage phagocytizes it, and then antigens from the bacteria that it killed end up on its surface. If a T helper cell comes along and its T cell receptor, right, can bind to that antigen, then it's activated. Now the important things, like, you know, when you take a test would be, you know, what's the antigen presenting cells? Is it MHC1? Is it MHC2? Right, know that CD4 is T helper, CD8, T cytotoxic. So keeping those straight is the most important thing. Don't worry too much about the details of activation, how that all happens, right? Unless you're a cell biologist, you know, I know it's much more complicated. You know, I won't get into the nitty-gritty details. So just to wrap up, right, B cells versus T cells, humoral and cell-mediated, those are the two main categories. And then on this table, don't worry too much. I usually like to just bring up the IgM, receptors for antigen is IgM and the T cell receptor. to emphasize that. And these are just a few other facts that I don't know if that's all that important. Now, adaptive or acquired, now I'm talking about acquired, which is adaptive. You can have natural adaptive or acquired, or artificial acquired. If it's natural, right, you got it through, well, how we've been getting it for eons. Artificial is a shot, right? It's a medical procedure. So I'll give you some examples. Then there's two types, two natural and two types of artificial, right? An active for either one means that the person is actually making their own immunity. Antibodies are usually what we use as examples for this. If it's passive for either one, then someone else made the antibody, you didn't. Could be your mother. Or you could take antibodies from another person and then inject them into you. So if you get natural immunity active, you've gotten it through, you caught it from somebody else, you made antibodies, T cells. If natural and say colostrum is natural, right, breast milk contains IgA, the secretory antibodies that get in you. They last for about a month or 2 months in the baby. They have a half-life, right? All proteins do. And also, any of mom's antibodies that cross the placenta during pregnancy, that's IgG, then the baby will have those for 2 months or so. Then artificial, say getting a flu shot or any vaccination, is active artificial, acquired. And then if you're given gamma globulin, that's an antibody. Anytime you're injected or you're getting a treatment that contains antibodies from another person, and I don't know if they do that with T cells, but you could. But technically, they probably wouldn't last too long, right? Because your body will try to reject them. So normally, this is always antibodies. So you can consider immunization, artificial, active, acquired. There are 5 types. I used to have messenger RNA on here, and I took it off, and then I put it back after COVID-19. So we're going to go through some of them. If you get the flu shot each year, this was from 2012, 2013, they call it a trivalent. That means sometimes now they have some with 4. That means they have, if it's tri, meaning 3, they have 3, like in this case, right, 3 strains of virus, and they're picking one of the antigens on the virus that they hope you make antibodies to. If you're given an inactivated whole agent, then you're given an inactive virus. The whole agent is the virus. And it won't multiply in your body. But what's good about it is that it contains the whole antigen, the whole animal. And that's always best when you're thinking about this. And attenuated means that you're given a virus that that'll actually multiply in your body, and that's probably the best, but it's more dangerous too. Now there is one for COVID-19 that I didn't list here, which is you're given an adenovirus that's not COVID-19, but it contains the virus. When it gets into your cells, will make COVID-19 spike protein. So maybe I should add that to this list too. That's kind of interesting. That's what's good about immunology. I don't know if everybody finds this stuff interesting, but most students do. Now, subunit would be my preferred, right? Even for COVID-19, I think the best thing to do would be use very small pieces of the COVID-19 spike protein, not the whole darn protein, and make specific antibodies, right, to the spike. So, you know, and you're only injecting the protein, which is what normally is found in vaccines, right? So if this is the spike protein, you might only need, you know, like say that part of the spike, and you inject that into somebody, and then they make antibodies to that portion of it. And instead of injecting, now with the messenger RNA that they've used, right, the mRNA viruses, instead of, when you get done with that, right, you inject the messenger RNA, and then you end up making the whole spike protein. So the tetanus, toxoid, I talked about this, that just means they inject an inactive toxin into you, so you make an antibody against a toxin if it ever got in your body. You'd have to be infected with the bacteria that makes the tetanus toxin, but that's the whole purpose. You know, this idea you need it every 7 years, that's just another, I don't know. I mean, you can argue in that case, you might not have a couple days for the antibodies to get into your bloodstream to neutralize it, so. But after you've gotten a few of these boosters, you tend to keep the antibody at a high level for a long time. But it's always different. How much antibody you make, how long you make it, it's very, you know, up to, you know, it's different for different people. Artificials, when you're given the antibody, right, like Rogam, we'll talk about what Rogam is, or an antivenom. Most of the time, you're given an antibody that was maybe generated in another animal. Gamma globulins are pooled serums. And then this is a list of, there's no questions from these tables, but I just add them